The Ambrose Laboratory researches various aspects of HIV from basic virology to animal models of infection to clinical samples. We are interested in how the virus infects cells, the evolution of drug resistance, how to improve HIV prophylaxis and treatment, and the establishment and evolution of viral reservoirs. Here are a couple of examples of the types of research we perform. We use cell culture to address some questions in the laboratory, such as what human cellular proteins HIV uses to infect cells. Our laboratory performs live cell imaging of HIV particles after entry into cells in collaboration with Pitt's Center for Biologic Imaging, or CBI. Here is an example of HIV particles containing fluorescently labeled viral integrase in red, trafficking on stained microtubules shown in white within a cell. We can track rapidly moving viral particles on their way to the nucleus of the cell. Using genetics and cell biology methods, we can make changes to the virus proteins or cellular factors to study how they affect HIV trafficking and infectivity in different cell types. In recent studies led by a former PMI PhD student, we tagged a cellular protein called CPSF6, which is known to bind to the HIV capsid using a green fluorescent protein, and we could visualize it associating with viral particles in the cell cytoplasm. Over time, the green CPSF6 protein dissociates from the HIV integrase signal, which is inside the capsid, suggesting that it can facilitate capsid disassembly. With live and fixed cell imaging, virology, and structural biology studies, we developed a model that suggests that during HIV infection of HeLa cells or CD4 positive T cells, cellular CPSF6 binds to HIV capsid in the cytoplasm near the nucleus and facilitates capsid uncoating, which is a highly regulated process. Another host protein called cyclophilin A, shown in orange, also binds to HIV capsid and is expressed at the periphery of these cells. Cyclophilin A protects capsid from binding to CPSF6 too early. However, in macrophages, cyclophilin A is more abundant than in HeLa and CD4 positive T cells and is expressed throughout the cytoplasm and the nucleus. This suggests that cyclophilin A is protecting the capsid from premature CPSF6 binding and may explain recent findings that capsid appears to be more intact in the nucleus of macrophages. Preventing CPSF6 or cyclophilin A binding to HIV capsid in macrophages leads to abnormal trafficking and infection. We are currently using super resolution imaging and correlative light electron microscopy to visualize HIV capsid entering the nucleus and studying the role of cytoskeletal proteins and the nuclear pore complexes that influence nuclear entry. Another recent study in the lab involves the use of humanized mice to study long-acting nanoparticle formulations of antiretroviral drugs. Nearly 2 million people are newly infected with HIV each year. Every week, about 6,000 young women become infected. In sub-Saharan Africa, 4 out of 5 new infections occur in girls between the ages of 15 to 19 years old. We need better HIV prevention methods, particularly for women. We have been studying long-acting formulations of antiretroviral drugs for therapy, but also for pre-exposure prophylaxis, or PrEP. This involves formulating an antiretroviral drug into nanoparticles with the hope of injecting people once a month or longer to protect them from HIV infection. This would improve adherence by eliminating the need to take daily PrEP pills. In a recent study, we asked the question whether long-acting nanoparticle PrEP could inhibit vaginal transmission of drug-resistant HIV. Because we cannot easily do these experiments in women, we used a mouse model in which the mice have a human immune system. We had one group of female mice that were given no treatment, and we had another group of mice that were given a single dose of long-acting PrEP. Then the mice were challenged vaginally with HIV. Using wild-type HIV, most of the mice that did not receive PrEP became infected within 3 weeks after virus challenge. However, the mice that received PrEP prior to HIV challenge were not infected as readily. By week 5 post-challenge, only 33% became infected, but a few mice had delayed transmission and infection. In contrast, using a mutant virus that had 25-fold resistance to the PrEP drug, nearly all untreated mice became infected, and all PrEP-treated mice also became infected, showing that a single drug in a nanoparticle formulation could not prevent infection with a moderately drug-resistant virus. And even when exposed to wild-type HIV, many of these female mice were not protected to a single long-acting PrEP dose. So we can do better. We are working with collaborators to test better antiretroviral drug delivery methods for both PrEP to prevent infection, particularly in women, as well as therapy to suppress virus in HIV-infected individuals. In addition, we are working with a PMI collaborator to understand HIV co-infection with Mycobacterium tuberculosis and how both pathogens influence each other, including evaluating HIV infection in granulomas in the lung, which can lead to worse outcomes for both infections. Similarly, we are developing a mouse model to study HIV co-infection with hepatitis B virus to again understand how each virus infection influences the other. To learn about our research, visit our website at www.ambrose-lab.com.